Okay, evolution is going to be the history of how these different species came about, starting from some ancestor DNA sequence or ancestor species, through the errors in the replication and other mechanisms that change the DNA and cause speciation events, how these different life forms come about. We're going to look at there are three different life forms that are sort of commonly accepted: bacteria, archaea, and eukaryotes. And generally, the bacteria and archaea are grouped into one. We'll just call them prokaryotes. Okay. Eukaryotes have a nucleus. They have a cell nucleus. They have different compartments within the cell, whereas prokaryotes don't have a nucleus. That's sort of their main separating feature. Different species vary in size. The small molecules and atoms are in the nanometer range. Proteins range from several hundred nanometers. Viruses are composed of some proteins and DNA complexes, DNA molecules. Prokaryotes are in the micrometer range, and eukaryotes are generally larger in the millimeter range. Okay, the largest cell is what? So an egg is also a single cell. It can be considered a single cell before it starts dividing, right? So an egg is also a eukaryotic cell, in a way. All right. So prokaryotes don't have any nucleus or organelles. They have single circular chromosomes. But in addition to the chromosomes, which are the DNA molecules, they also have additional DNA molecules called plasmids. They are smaller. They provide drug resistance and rare compound metabolism. And these plasmids can be interchanged between different bacteria. So one bacteria can sort of give it to the environment, and then it can be taken up by another bacteria, or they can sort of have a fusion or sort of interchange through different mechanisms. So the drug resistance and rare compound metabolism can be obtained from another bacteria. The most studied prokaryotic organism is E. coli, that is mostly present in the gut. Eukaryotic cells are animal cells, plant cells, fungi, and algae, and so on. They are compartmentalized. Unlike the prokaryotic cells, they have organelles. These compartments are nucleus, mitochondria, and chloroplast. So according to the evolutionary sort of hypothesis, there was one prokaryotic cell that sort of engulfed one or more other prokaryotic cells, which ended up being compartments within the larger prokaryotic cell. A nucleus is sort of a different life form that got engulfed by a prokaryote, and then it started replicating within that host cell. Okay. All right. So mitochondria is sort of in a way, infection of the cell, if you think about it that way. All right. Eukaryotes have multiple linear chromosomes. Humans have how many? 23 pairs, right? So we have multiple linear chromosomes. Prokaryotes have single circular DNA molecules, whereas eukaryotes have multiple linear chromosomes. And sometimes we have eukaryotes can have more than one copy. So humans have 2 copies of each chromosome, but plant cells can have many more copies of each chromosome. Okay. A picture of a typical eukaryotic cell is shown here. Let's take a look at it and go over different parts of this cell. So we have the nucleus here, which contains the chromosomes. There's a part of the nucleus called nucleolus, which is sort of a little dark spot where transcription goes on. So there are several enzymes. There are some enzymes there that sort of drive the transcription. Transcription can actually go on anywhere else within the nucleus, but that's sort of a highly concentrated area where the transcription happens. Okay. We have ribosomes, which are these little dots. They can be bound to this membrane structure, or they can also be found swimming around in the cell. Okay. Ribosomes are responsible for converting RNA to protein, which is a translation. So the RNA molecule is translated into a protein molecule. The ones that are bound to the endoplasmic reticulum, so this is this membrane structure, make up a rough endoplasmic reticulum. So if an endoplasmic reticulum has ribosomes bound to it, we call it rough. If it doesn't, we call it a smooth endoplasmic reticulum. Okay. As the let's say this is an ER, this is a ribosome. As the RNA molecule is transcribed into a protein, it's translated into a protein. That protein can sort of enter the endoplasmic reticulum and get further processed within this membrane structure. Okay, so it's being translated into a protein, and the protein that's being produced is sort of sucked into the endoplasmic reticulum as it's being produced, and it's channeled into different post-translational modifications as it goes through this endoplasmic reticulum and gets into the Golgi structure, and then the protein can then either get into the cytoplasm by coming out of the endoplasmic reticulum, or it could be excreted from the cell by being packaged into these vesicles that then go and sort of merge into the cell membrane and then come open. Everything that was inside the vesicle will then be released to the environment. Okay. The ER, the endoplasmic reticulum, and the Golgi, they are membrane structures just like the cell membrane. And there's you can have a portion of this sort of come out and then bind to the cell membrane, or go the other way. So that if you have buddings of the cell membrane, it can go and bind to other membrane structures within the cell. Okay? That's how some of the molecules are being exchanged by these different compartments. Where's 6? So this is the Golgi structure where protein post-translational modifications take place. And then we have the cytoskeleton. You just wouldn't have one only cytoskeleton. There would be many cytoskeletal structures that create sort of a skeleton to the cell. It provides the shape to the cell, and sometimes it also provides mobility to the cell. Some cells can move, right? That movement is accomplished by their internal cytoskeleton. Right? The cytoskeleton can sort of move on top of each other using different motor molecules, which will then shift the entire cell forward in a certain direction. Okay, so what was the other one? Right, mitochondria are these shapes. Mitochondria have their own DNA molecule. They're just like our chromosome. Here, this is also another membrane structure. It's separating itself from its environment, and then within this mitochondria we have DNA. DNA replication takes place within the mitochondria as well. So mitochondria replicates itself, but because it has lived within the host cell for many years, right? It didn't. Over the evolution, it lost some of its genetic makeup because a lot of the proteins that are necessary for mitochondrial function are now provided by the host cell. It didn't have to maintain those genetic sequences. So it lost its genetic makeup because of its dependency now on the host cell. Okay. It's a minimal set of proteins that it transcribes, and a lot of the other proteins that are needed for mitochondrial function are sort of transferred from the host into the mitochondria. Chloroplast is a similar structure that has its own DNA, and it can also replicate within the cell. Cytoplasm is everything inside the cell. It's called a cytoplasm. Lysosomes are these small vesicles, and they have reactions taking place in them, usually reactions that sort of break things apart. So if you have old proteins or old RNA molecules that are no longer used, they are targeted into lysosomes and they break apart back into its monomers. So we don't just build, we also break apart the things that we build as they age. Okay. All right. And that's it. Any questions about these different compartments? Okay. Let's look at the transcription, which is the process of converting a DNA molecule to an RNA molecule. And that is accomplished using by the RNA polymerase enzyme. So this large blue animal is the RNA polymerase. Okay. It goes and binds to the DNA, and it unwinds a portion of the DNA, and the template is then used to produce a new RNA molecule. Okay, so this newly synthesized RNA transcript is being produced monomer by monomer, nucleotide by nucleotide, as the RNA polymerase is sort of like a zipper action as it is going through the DNA. Okay? It unwinds from one end, it rewinds from the other end as it is done transcription. Okay. All right. There are some terms that are going to be important to remember. The DNA is a double helix, right? It's a double-stranded DNA. We'll call one strand the coding strand, and the other strand is going to be the non-coding strand. The non-coding strand is what physically binds to the RNA that's being produced. So this one right here is the non-coding strand. Okay, the coding strand is not used as sort of hanging out here. But because of the complementarity of the RNA polymerase, RNA transcription, the nucleotides that are being produced on the RNA will be complementary to the non-coding strand. Okay, so if there's a G on the non-coding strand, the RNA molecule that's being produced will have a C corresponding to that G. If there is a T, if there's an A instead of a thymine, now we'll have a uracil. Okay, so with RNAs, we'll just use the letter U instead of the letter T. The non-coding strand and the transcribed RNA are complementary to each other. But if you look at the RNA molecule, the sequence of the RNA molecule, and the coding strand, which is a strand that is not being used physically, they will be identical except for the T to U conversion. Make sense? So if you call this X, Y is complementary. This is also Y, let's call it Y prime because of the uracil 2 thymine. This is complementary, that's complementary, therefore these two should be the same. Make sense? All right. Okay, so then the coding strand is identical to the mRNA molecule that's being produced, but the coding strand is not a physical template. There's no physical interaction with the RNA being produced. Yes.
So when you do the specifile in terms of the coding strand, right?
Yes. Okay. Mm-hmm. So by convention, we always give a DNA molecule starting from its 5' end to 3' end. So when I say I have a DNA molecule that is ACTA, implicitly, I'm saying that this is the 5' end, and this is the 3' end. Okay. All right. So we have a really long chromosome, right, or a really long prokaryotic circular DNA molecule. Portions of this long DNA molecule will be genes. There are going to be many thousands of these smaller chunks of the DNA that form a single gene, that each form a gene. These genes will have a certain structure in terms of different parts that are specific. Prokaryotic genes and eukaryotic genes will have different structures. A prokaryotic gene has a transcription start site. So I'm looking at a DNA molecule right here, a circular DNA. There's one gene here. Okay. There may be another gene somewhere else. The transcription start site is a particular set of nucleotide sequences, say it's AT-ACT. So that is a sort of a signal for the RNA polymerase molecule to start its transcription process. So when the RNA polymerase binds to DNA, it doesn't just start transcribing. It will first scan. Once it hits this transcription start site, then it will start producing the RNA molecule. Make sense? Upstream to this transcription start site, we have regions of activators and repressors. What that means is there are other portions that come before the transcription start site that sort of change the efficacy of transcription. If you have certain molecules that bind to these regions, called transcription factors or repressors, they will affect the RNA polymerase binding there. So they may block it from binding, or they may enhance its binding. Therefore, they can change the amount by which, or the speed at which, this gene is being transcribed into an RNA molecule. Right? The transcription factors control gene expression in that way. If you have more of this protein right here that sort of, say, blocks this transcription, and you're going to have many copies of this repressor protein that goes and binds there, and the more you have it, the less likely the RNA polymerase molecule will be able to start the transcription there, and you will have less of this mRNA molecule being produced. Okay. All right. Once the transcription start site is recognized and we produce the mRNA, it'll have to stop at some point, and that again is a particular signal that ends with AAA, and it also has a characteristic that forms a hairpin loop. Okay, what that means is the going that way and that way, we will have complementary nucleotides. So let's say I have A, U, A, U, or let's make that a G. Going the other way, I have U, A, U, C. If you open it up, I have A, U, A, G, C, U, A, U. All right, so this is the sequence here on the coding strand, and this would then be a signal sort of to form a hairpin loop, because one half of it is complementary to the other half. If you fold it over, it forms this structure, and this sort of clips in a way the transcription process. The RNA polymerase falls off and stops transcribing that portion of the DNA molecule. Make sense? A lot of these processes are sort of physical interactions, and sequence complementarity or sequence signals drive what happens where it happens. All right. And again, this is the anti-coding strand that provides the physical template. The coding strand that is complementary to this anti-coding strand will be identical in sequence to the mRNA molecule. Okay, so that was the prokaryotic gene. Eukaryotic gene is slightly more complex. It has a different structure and sort of different conventions because now we have different enzymes working in the cell. And these different enzymes will detect different signals on the DNA molecule. We again have transcription start site that sort of signals the start of the transcription for the RNA polymerase molecule. There's a portion called Tata box that is rich in TA and TA nucleotides. This region right here will be the region containing core promoters. A promoter is a place on the DNA that controls transcription. So these enhancers and repressors are sort of our promoter regions. Unlike the prokaryotic gene, the eukaryotic enhancers and suppressors don't have to be in close proximity to the gene. They can be many thousands of bases, thousands of nucleotides upstream to the transcription start site. Okay. And the way that they are able to control the transcription is because DNA molecule isn't just one long linear string. It sort of folds back onto itself. It's wrapped on histone molecules. So as a result, this portion of the DNA would be then spatially in close proximity to this portion of the DNA. Right? So if I have, let's say this is my enhancer repressor region, many thousands of base pairs, bases, and then this is my transcription start site. Okay. All right. And again, enhancers and repressors are regions in the DNA that are recognized by different proteins that either repress or activate transcription of that gene. The end of the mRNA molecule that's being produced has different signals as well. We have a poly A tail. That is a sequence of adenines that are added onto the mRNA strand after it has been transcribed. There are other signals, CA or GT in this case, if you're looking at the anti-coding strand, and adenine-rich region that is usually at the end of the transcribed mRNA molecule. Okay. So this mRNA molecule will not be translated into protein in its entirety. Just like from the DNA molecule, we have a portion of it that is being transcribed. From the mRNA molecule, we will also have a portion of it that will be translated into a protein. Okay, so the processes that I've described in terms of where things start and where things end, and how things are controlled, are going to be again available when we go from mRNA to protein. Okay. But now the molecule that we are operating on is going to be this mRNA molecule. A portion of it will be recognized as translation start site, and a portion will be the translation stop site. Okay. All right. In eukaryotic genes, we have post-transcriptional modifications that the prokaryotic genes don't have. What that means is after the RNA polymerase molecule has transcribed a DNA into RNA, that RNA molecule gets some additional modifications performed on top of it. RNA capping is one of them, where you just get one special nucleotide. It's not one of the 4 letters that we have, it's something else. It provides sort of stability to the RNA molecule so it doesn't get chopped up by the enzymes in the cell. It's sort of like protection cap that we have on the RNA. The polyadenylation is done at the tail end of the RNA molecule. Where's my color? Okay, so this is the transcription start site. And then once it's transcribed, we're going to get a cap on it. And then this is the poly A site, where we get polyadenylation, where about 200 adenines are added to the 3' end of the RNA. We also get RNA splicing to remove introns. So with eukaryotic genes, the RNA molecule that we produced will have different domains. These domains are called exons, and everything in between, introns, are removed from the RNA molecule. If you have, say, 3 exons in an RNA molecule that are, and two introns, we have something called alternative splicing. So what could happen is we could remove this one and that one separately, and we'll end up with exon 1, 2, 3 in the final mRNA molecule. Or we could match this end with that end and remove this portion entirely, which will result in domains only 1 and 3 being produced as the mRNA. And when you use domains 1 and 3, you're going to end up with a different protein product. When you use domains 1, 2, and 3, which will be sort of a protein chain, a protein portion of the protein, and sort of a different portion that's linking the 2, 1, and 3. The alternative splicing gives rise to different protein products. So we have one gene, but now because of this shuffling, many different ways of removing the intronic regions, we end up with different possibilities. Make sense? Okay. The process by which introns are removed is shown on this slide. So one end of the exon and the beginning of another exon are paired up. And this pairing results in a loop structure, which is then removed, and there's an enzyme that links to the 2 ends of the exons. And this loop structure has a special name called lariat. It's the thing that the cowboys throw. Okay, lariat. Looks like that. Okay. Let's look at an open reading frame. So we've produced our mRNA molecule. Now we are ready to generate a protein from it. The way that we produce a protein is a ribosome will bind to the mRNA molecule, and it will sort of read out the sequence of nucleotides that are present on that mRNA molecule. And depending on what nucleotides you have on the mRNA, a different amino acid will be linked to the protein sequence that we are growing. So with DNA replication and transcription, things were slightly easy, right? We just had a 1-to-1 correspondence. If there's an A, we just produce a T. If there's a C, we produce a G. There's a 1-to-1 complementarity in the way that the RNA polymerase produces a new RNA molecule. With ribosome, we don't have that anymore, right? So there's not going to be a 1-to-1 correspondence between a single nucleotide and a single amino acid. So we are talking about two different molecules now. We are trying to convert nucleotides to amino acids. And nucleotides and amino acids don't have a complementarity relationship, so we cannot really make use of any complementarity that we've made use of when we did DNA replication or RNA transcription. So one of the constraints for translation is that there are 4 different nucleotides. but there are, say, in the order of 20 different naturally occurring amino acids. There are actually more, but we'll just say 20 is the commonly accepted number. All right. So we need a different mapping. We cannot, if we did 1-to-1, we would only be able to make use of 4 amino acids. So what the cell has sort of invented is to use triplets of nucleotides to encode different types of amino acids. Okay. So what we'll do is we'll say a CAT will be a particular amino acid, and ACT will encode a different amino acid. So in a way, we have, say, an amino acid alanine. Let's say histidine. Bad writing. Let me rewrite that. CAT will encode a histidine. There's going to be a histidine amino acid that sort of correspond to a CAT. Whenever we see a CAT, the ribosome will produce a histidine. And this correspondence is not done by binding, by having histidine being directly complementary to CAT. There's going to be some sort of adapter molecules that manage this complementarity. And these adapter molecules are called transfer RNAs, as we will see in the next few slides. So what we'll do is we'll take a DNA and messenger RNA, we're going to have to chunk it into triplets of nucleotides. We could start with CAT, and it will give us CAT, ACT, and so on. Or we could skip the C and start with ATA and convert into triplets of nucleotides that way. Or we could skip C and A and start with TAC. Depending on where we start, we get a different set of triplets, right? And these different starting positions will give us what we call different frames. Frame 0 is going to be where we start at the 0 index. Frame 1 will be where we skip the first one. Frame 2 is where we skip the first 2. Make sense? Okay. Could you close the door for us? Thank you. So some of these triplets of codons have stars on them. Any ideas why I've marked them specially? Yeah, so these are stop codons. When the ribosome moves over the, let me think about this, moves over which way? For you guys, it'll be this way. When the ribosome moves over the RNA molecule, once it hits a certain codon, certain triplet of nucleotides, that'll be the end of the translation. Just like with the RNA polymerase, when it hits a certain sort of loop structure that marks the end. So here with the ribosome, where we do translation from RNA to protein, a triplet of nucleotides will mark the end of the translation. Okay? And very different signals. TAA and TAG are 2 of them. There's a third one. So whenever the ribosome hits a TAA, it will stop translating the molecule. It will stop translating the protein. Okay. So the ribosome will have to sort of grab, in a way, triplets of nucleotides, detect what it is. If it's a stop codon, it'll stop translation. So the ribosome will have to sort of slide over the RNA molecule in triplets of nucleotides. Okay, so different frames will give rise to different size proteins. Frame 0, let's say the ribosome binds here and produces one amino acid from that, another amino acid from that, another amino acid from that. As a result, we get a protein that is 3 amino acids long, because it will stop right here, right? With frame 1 is when the ribosome skips that first C and starts its detection with the next triplet, ATA and so on. We don't hit any stop signals. There may be stop signals beyond here after this sequence. So this protein right here will be 1, 2, 3, 4, 8 amino acids long. And the next one right here is going to be just 1 amino acid long because this is the stop signal. All right. So generally, the assumption is that a short protein doesn't make sense, right? If you see a long open reading frame, that is less likely to be there by chance. Therefore, the frame with the longest protein will be used as an open reading frame, will be the one that we assume will produce a protein. Out of these 3 different possibilities, we'll pick the one that gives us the longest protein. Make sense? Okay. So actually, not just 3. When you encounter a DNA sequence that is given to you from 5' to 3', you consider 3 frames in one direction, but then you also have to consider the 3 frames from the other direction, because the RNA polymerase could use either one as coding or non-coding. All right. So when we encounter a DNA sequence, we have to look at both directions to see what proteins can form from either reading frame. So we have 6 reading frames that we have to consider for each DNA sequence that we encounter, and then we'll use the largest one, the one that gives us the largest protein, and consider that as the open reading frame. Okay. All right. Open reading frames are not necessarily independent, especially in viral genomes. You can have overlapping open reading frames. So a portion of the viral genome can be one gene, right? And a portion of that gene can, going in the other direction, can give you a different gene. All right. So viral genomes are highly efficient in terms of their size, and they accomplish that by having these overlapping open reading frames where if you go one way, you produce one gene. If you go another way, using the same DNA, either coding or the non-coding, you produce another gene, another protein. Okay. In bacterial genomes, we have structures called operons, where the transcription of many different genes are orchestrated using the same transcription start site and the same promoter regions. Okay. So what we have, this is an RNA molecule where the ribosome can bind, this is messenger RNA, where the ribosome can start processing at different positions. So different ribosomes can do their own thing and produce different proteins. At the DNA level, we have the RNA polymerase will slide over this DNA molecule and produce this messenger RNA that contains different genes on it. Okay? So we have one messenger RNA that can produce many different proteins that are located at different positions on this messenger RNA. So what would be the advantage of this? Any suggestions? So why should we have operons? Why not just produce this one as a separate messenger RNA, that one as a separate messenger RNA, and this one as a separate messenger RNA? You said error? It would be more error prone? Not quite. Yes. How so? So then why not just transcribe the entire genome? That would be the ultimate efficiency in that, if that's the case, right? They don't transcribe the entire genome. They only transcribe the portions that they need.
those proteins apply in the same process.
Right. So if you look at a pathway, where was my pathway? This one right here. There's going to be an enzyme, a protein that is driving this reaction. There's going to be another protein that's driving this other reaction. To produce these proteins at different amounts, there's not just one reaction going on, there's 1,000 copies of this reaction going on in the cell. The cell has to produce this protein right here. The cell also has to produce this protein right there. If you have proteins that are functionally related in terms of their copies, it makes sense to produce them in similar amounts. And for those functionally related proteins, bacterial genomes, the prokaryotic genomes, places them on operons so that these functionally related proteins get transcribed at the same time. You are more likely to have a similar amount of these proteins in the cell, where you don't redundantly create many copies of this one, and the next one becomes a bottleneck. It doesn't matter how much of this product you have right here. If you don't have enough of this other protein, you're not going to be able to drive this entire pathway in a very efficient manner. Okay? So functionally related proteins are placed in an operon so that they are produced in similar copies in the cell. This is more so in prokaryotic genes. In eukaryotic genes, we usually have one messenger RNA to one protein product, except now we have alternative splicing where different portions of this messenger RNA can be spliced out and fused together to produce a different protein product. Okay. So you may have a protein that's composed of different domains that acts in different pathways, and you can manage that by plugging and placing different domains within that. Different combinations of domains will result in different functionalities. All right. The triplet of nucleotides is called a codon. So when we chunk out the messenger RNA into triplets, we'll call each one a codon. We have the coding sequence, which is the portion of the messenger RNA that results in a protein. So when we produce the messenger RNA, not the entire messenger RNA is going to become a protein. There's going to be a translation start site, translation stop signal. Only that portion will be the coding sequence. Okay. 5' UTR is 5' untranslated region, meaning that portion does not become a protein. 3' untranslated region is again not translated into a protein. We have that triplet stop signal and a start signal. In between that is a sequence that will be converted into a protein. Okay. And this figure shows 3 different frames. Depending on how you chunk out these different frames, you're going to end up with different amino acid sequences. The translation initiation signals are different in bacteria and eukaryotes, just like the transcription initiation signals were slightly different. In bacteria, we have Shine-Dalgarno sequence, which is a consensus AGG, AGGU. So this appears upstream of an AUG nucleotide, which is the start nucleotide. The translation start site is an AUG. When the ribosome hits an AUG, that's where it starts producing a protein. Okay, so an AUG will produce a methionine amino acid. Before this AUG, we have this consensus sequence that the ribosome recognizes. The ribosome will come and bind to this Shine-Dalgarno sequence, and then sort of start scanning. Actually, it's very close. It doesn't even have to scan, and it'll start translating from the AUG. Okay. With eukaryotes, we again have we have a different process. We don't have a consensus sequence. Instead, the ribosome will bind at the 5' end, that special cap, which is this special nucleotide, and it'll just start scanning until it finds an AUG. Okay. Once it finds an AUG, then it'll start producing the protein from that. Make sense? Okay. The translation itself is described on the slide. The ribosome shown in green is composed of different subunits. It's composed of different proteins that come together to produce this machinery. Okay. The small subunit and the large subunit together hold sort of 3 different pockets. The A side, which is the acceptor side, is where a transfer RNA comes and binds. Elongation site, and I don't know what P stands for. So these 3 different pockets hold transfer RNAs as the protein is being produced. Okay. The messenger RNA, as it is sliding through these different pockets, when it is in the acceptor site, a particular messenger RNA that has nucleotides complementary to that codon will come and bind there, and it will be holding a particular amino acid. Okay, and that's described as a genetic code. So if my right arm is a tRNA, and this tRNA is complementary to UUU, it would be AAA, right? Then this right arm will have to carry a phenylalanine, this particular amino acid. Okay. And then when when the ribosome has a UUU codon, AAA will match, and this transfer RNA is holding a phenylalanine, which is going to be attached to a growing chain of protein.